We encounter a number of challenges in relation to environmental factors in this project. Some of them included drought, which was frequently occurring in some of these environments, particularly East Kadem, which affected some of the demonstration plots.
To wapuro to kae to chieng' orieny molalo cham ma cham oko wayude e yo maber kendo ka giti to giti mongelore.
Mokwongo nwatemo wapidho laini moulo ketho. Koro achien to koro walude kama akinyaluo kaka yande de waludo. Ema dhi ogolo nusegi. Koro magi bi oula moywero dhi otimne mochokore. E tabu manche wan go. Ema wayude wach pur.
We also had a problem with storm and lodging. We had serious problems of lodging, particularly when finger millet was well taken care of and we provided nitrogen. Also, a major key challenge that we realized on farm was the presence of striga weed, this purple-flowered Striga harmontica, which is almost a major problem in the whole of Western Kenya where we grow finger millet.
Wan gi buya mar kayongo ene mosebet ko chando wa puothe makata kipuro to kiyud keyo maber nikech buyani. Koro kaka jo molony kuom agriculture wadwa nu konywa gima wanyalo daro go kayongo puodho mondo wayud chiemo maber nikech watiyo mang'eny to harvest matin nikech kayongo. Thungo chamo tamo gidongo ka to githirno kar dongo mondo giket wiye gi madongo to githirno koro gin mana gik mong'wony mok dongo kendo wigi gigolo mathindo thindo.
There are a number of outputs that this project achieved in the end. This is seed multiplication of the major output of the project. We have selected white-seeded finger millet that is lodging resistant. This is a major problem in finger millet production because if we give finger millet nitrogen, most of it would lodge, would fall over. But with this new selection, even with high levels of nitrogen, it would just grow and convert that into grains, which means we can actually almost double the yield with this kind of variety compared to the traditional ones that would fall over or lodge and get destroyed in the process. So this is a key output of the project that we are multiplying the seed. So now farmers have access to these new varieties and improved seed that they can use to improve their finger millet production or productivity.
No ayude yuto maber kabisa. Kendo no konyowa, kendo no miyo wategno e girubwa na mang'eny.
Kendo ne watiyo maber, mane wakeyo, mane wanwang'o gorogoro 40
Chiro hie to en go. Kendo sana koth cham mano miyo wani en kodhi maber.
Kal nigi pesa, moingo ene adha ginen. Oingo oduma gini, nikech oduma, gorogoro achiel, biro bet siling 20, kata 25 sama piny ochiek. To kal ok bidhi less than 200 gorogoro achiel. Which means nango en gi yuto moingo kothe ma wan go gite.
Another output, key output of this project was we identified some very high yielding finger millet selections that are early maturing. Some of them are even resistant to striga weed.
Wamor gi kodhi mencha ne Professor Kelonwa koa Maseno University. Kaluwore ni kothe manwa komo chon, ok gitim maber ahinya, giruyo chiek to wigi ok dongo.
Kal ma pidho kani en sani ko dong weeks 2 takae. Kendo onyak mang'eny to tieko ndalo matin. Kwa ng'iyo ma choncha ne tieko dweche 6 to ma tieko dweche 3 to oseikre. Koro dhi piyo to kendo nyak mang'eny.
Masani mokelgi iyudo ni nigi gik mang'eny monego kony dhano del molo kothe man wapidho machon. Nikech nitie gik momedie value addition momede kothego. Mamiyo kaka watiyo kodgi to gibeyo molo man watiyo godo chon.
This finger millet project went for only 2 years and we were unable to cover other diverse areas of finger millet growing, particularly the Rift Valley region or Witen area, which is a major finger millet growing area, but we were unable to engage with farmers. So I think in future, if there's availability of funds, we would engage those farmers and we would move on to a wider area so that we give them new varieties, we multiply these new varieties that are better and avail them to farmers so that they can have improved finger millet productivity, improved income, and generally improved livelihood. This project was funded by FAO, Benefit Sharing Funds of the treaty, and we are very thankful and grateful to FAO, that is Food and Agriculture Organization, or Benefit Sharing Fund, for giving us money to undertake these activities over the last 2 years. We are very grateful, and we hope some of the intended objectives of this project have been achieved through the generous support from this organization. We would also like to thank Maseno University that has managed this project all through and provided enabling environment for this project to flourish. Particularly, we have used the Maseno University land for seed multiplication, for agronomic evaluation and everything, and they have given us every support. So we also like to thank Maseno University for giving us this support and managing funds for this project. In this project, we are also partnering with the Kari Gene Bank, Kenya Agricultural Research Institute, who would be the eventual keepers of some of these materials that we have developed. So we would like to thank the Kari Gene Bank for being good partners in this project, and some of the outputs of this project, they would be the eventual, we'll deposit some of these new materials with the Gene Bank for wide availability to the scientific community, not only in this country but worldwide.